Welcome back. In this video, we're going to talk about antibodies. And I want to start with one simple idea. Antibodies are powerful, but they're not magic. They do not just float around and vaguely fight infection. They work because their structure gives them specific jobs. By the end of this short video, you'll be able to look at a problem and ask, what does the antibody need to do here? Does it need to block entry, help something get eaten, trigger complement, or mark a cell for killing? That's the whole goal. Hi, I'm Christopher, and in this video, the big idea is that antibodies have two major working regions. The FAB region is the recognition end. That is the part that binds target. It recognizes a specific antigen, or more precisely, a specific epitope on that antigen. The FC region is the recruitment end. It acts like an immune system handle. Once the FAB has found the target, the FC region helps recruit the response. A simple way to remember this is FAB finds, FC calls for help. Let's start with FAB. FAB stands for antigen binding fragment, and for our purposes, think of it as a specific recognition end of the antibody. The FAB region binds a specific epitope. That specificity matters because the immune system is not trying to bind everything. It is trying to bind the right thing. When FAB binds to a viral surface protein, for example, it may physically block the virus from attaching to a host cell receptor. If the pathogen cannot attach, it often cannot enter. That is the setup for neutralization. Now let's give FC its moment. The C in FC does not stand for constant, although that would be a very reasonable association. FC stands for fragment crystallizable. Functionally, though, the FC region is built from the constant regions of the heavy chains, and those constant regions help determine the antibody's class-related effector functions. But here's the key point. FC is not the antigen binding end. After the FAB region binds the target, the FC region faces outward. That exposed FC region acts like an immune system handle. It can interact with FC receptors on immune cells, help activate complement, and influence what kind of effector response the antibody can support. FC is incredibly important. FAB recognizes the target. FC helps turn recognition into action. The first big antibody job is neutralization. This is usually the best answer when the problem is a virus, toxin, or other agent that needs to bind something in order to enter or damage the host. With a virus, neutralizing antibodies bind surface antigens and physically block attachment or entry by the virus. That is mostly an FAB job. The antibody protects by getting in the pathogen's way. That is why neutralization is especially important before infection is well established. It's a pre-entry strategy. It stops the next rounds of cells from being infected. The second big job is opsonization, which is a fancy word for tagging something so phagocytes can recognize and eat it more efficiently. This is a great example of FAB and FC working together. FAB binds the bacterial surface antigen. The FC region points outward away from the bacterium like a handle. Phagocytes have FC receptors and can grab that handle. The antibody is making the bacterium easier for a phagocyte to find, grab, and internalize. The third job is complement activation. When antibodies, especially IgM or certain IgG subclasses, bind to a pathogen surface, their FC regions can help recruit complement proteins such as C1q. That can trigger the classical complement pathway. Complement can help destroy pathogens, coat them for better phagocytosis, recruit inflammatory cells, and improve clearance. This is also where we start to see the danger of powerful immune tools. Complement is useful when controlled and targeted. If it is excessive, misplaced, or poorly regulated, the same inflammatory power can contribute to tissue damage. The fourth job is ADCC, or antibody-dependent cellular cytotoxicity. The key idea here is that the target is a cell. not a free-floating virus, and not a bacterium waiting to be eaten. In ADCC, antibodies bind antigen displayed on the surface of an infected or abnormal host cell. The Fc region of that antibody is then recognized by Fc receptors on an effector cell, classically a natural killer or NK cell. The effector cell is activated and kills the antibody-coated target cell. So ADCC is about eliminating a marked cell. That distinction helps separate ADCC from neutralization. Now we can match the antibody job to the problem. If a virus needs to enter a host cell, the most useful antibody mechanism is usually neutralization. If an extracellular bacterium needs to be eaten, opsonization makes sense. If an antibody-coated pathogen surface can trigger a cascade, complement activation may help with destruction, inflammation, and clearance. And if an infected cell is displaying target antigen on its surface, ADCC may be the best fit. Notice the pattern. The best mechanism depends on what the pathogen is doing and where it is located. Here's the take-home message. FAB finds, FC calls for help. Function depends on the problem. Antibodies are not generic magic molecules, and the best response is not always simply stronger immunity. Stronger in the wrong place can become damaging. Instead, ask 3 questions. What is the pathogen doing? Where is it located? And what kind of immune help is needed? If you can answer those questions, you can usually predict which antibody function matters most: neutralization, opsonization, complement activation, or ADCC. Thanks for watching. I'm Christopher Lee. What you understand today helps you protect patients tomorrow. And I'll see you in the next video.